All right, so let's take a look at macromolecules, otherwise known as large molecules. Macro means large. Well, there are four important biological macromolecules. Those are carbs, lipids, proteins, and nucleic acids. Okay, let's take a look at carbs first, otherwise known as carbohydrates, otherwise known as saccharides. Now, you can have simple carbs like mono and disaccharides. Mono means one, di means two. We'll see what that means in a minute. Those are also called monomers. Monomers are building blocks. So simple carbs are building blocks versus complex carbs, complex carbohydrates, are also known as polysaccharides, also known as polymers, large molecules. Okay, so here we have a couple monosaccharides. These are each simple carbs: glucose, fructose, galactose, deoxyribose, and ribose. Okay, each of these here up top has the chemical formula C6H12O6. There are 6 carbons, 12 hydrogens, and 6 oxygens in each of them. Those are simple carbs, monosaccharides. These folks here are called disaccharides because there are 2 monosaccharides or 2 monomers stuck together. How do they get stuck together? By something called dehydration synthesis. What we do is we take an OH off this guy and we take an H off this guy, and we're left with an O in the middle. Well, when you take an OH off one and you take an H off the other, they form water. So if you take water away from these 2 molecules, you're dehydrating them, and forming a disaccharide with an oxygen in the middle. So each of these 3 guys here are disaccharides, and they're simple carbs because they're not very, very long chains. However, these 3 here, these 4 here rather, are what we call polysaccharides. They're complex carbs. They can either be chains or they can be like fibers, for example. All right, examples of those would be like amylose, starch, glycogen, cellulose. Now, interestingly enough, we as humans cannot digest cellulose. There is the bond between those monomers, between those simple sugars, we can't break. So herbivores like cows, they can manage to break down those bonds, but we do not. So whatever we cannot break down, we can't digest, we can't absorb, passes through us. So otherwise known as fiber. So cellulose is what plant cell walls are made out of. So if you want to keep your diet regular and not get plugged up, eat lots of vegetables. Lipids, otherwise known as fats and oils. Okay, so basically what lipids are, it's a molecule of something called glycerol attached to, could be a variable amount, but for triglycerides, they're attached to 3 what we call fatty acids. So this is what we call a triglyceride here. It's a glycerol molecule bonded to 3 fatty acids. Now, if those fatty acids are straight chains here, we call them saturated fatty acids. You can think of it as parking spots here. Every available parking spot is filled with a hydrogen. They are saturated. This guy here is an unsaturated fatty acid. There are 2 open parking spots. Those fatty acids are unsaturated, which means that they don't pack together really nicely. So unsaturated fatty acids are liquids at room temperature, like oil, vegetable oil, canola oil, palm oil, olive oil, okay? Whereas saturated fatty acids are solids at room temperature because they pack together very nicely. And those are what we call fats, like animal fats. Okay? Proteins. Proteins are made up of a combination of 20 known amino acids. Just as there are 26 letters in the English alphabet, we can make hundreds of thousands of words from those 26 letters. Well, we can make lots and lots of proteins from 20 amino acids. So they all have the same basic structure. The only thing different between the 20 amino acids is what we call this R group or side chain. Here is a chart of all the different amino acids, and we can see they all have the same structure up top, but hanging down, highlighted in blue, are what we call the R groups or side chains, and that's what gives each individual amino acid different properties. Some of them are polar, some of them are non-polar, and on and on. Well, same deal. If we take an OH off this guy and an H off this guy, we make what's called a peptide bond. So we take a monomer and a monomer and we turn it into a dimer. So we take a single amino acid and a single amino acid and we bond them together and we're starting to build a chain. That's what we call a peptide bond. Well, if we take that chain and we make it longer, and we fold it up, then we have a protein. You might remember this from biology. I know it's been a couple years for some of you. Here's a cell, here's the nucleus, here is the rough VR. We know it's rough because it has ribosomes on it, and ribosomes are the location for protein synthesis. Okay, this is what we call gene expression, transcription, and translation. I know I'm bringing up some pretty far away memories here. So in a nucleus we've got DNA. DNA unwinds, unzips, and we make a copy of it called messenger RNA. That messenger RNA leaves the nucleus, goes into the endoplasmic reticulum, and goes to a ribosome, and that is where the ribosome reads the messenger RNA, and up until that point that was called transcription. But now that the ribosome is reading the messenger RNA, we call that translation, and based on the code that is in the messenger RNA that came from your DNA, that code tells the ribosome what amino acid to attach on next. So it forms an elongating chain of amino acids, which then pass into the Golgi bodies, and the Golgi bodies, if you remember, are kind of like the post office. They make sure that the postage, you have enough stamps on there, and that you have the right address. And if it's a correct, done correctly, then we have a protein. So that's how we make proteins in our body: gene expression, transcription, translation. Well, these are some take that back. One more section here. All right, nucleic acids, DNA and RNA. They don't just come out of nowhere, okay? So here's DNA. It's a double-stranded, it's a double helix, okay? A's and T's and C's and G's. Hopefully you remember that. Well, those are made up of sugars. It's either deoxyribose or ribose. The D is in DNA is deoxyribose. The R in RNA is ribose, OSE, and so lots of sugars and an OSE. So you have a sugar, you have a phosphate. So remember, sugar, phosphate, backbone. Sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay? And attached to those sugars is one of the bases: an A, a T, a C, a G, or a U if we're talking about RNA. Okay? So basically anything that you eat that's living, which hopefully you don't, well, yeah, I guess when you eat like salt, that's not living. But when you eat food like meat, when you eat vegetables, those were living materials, those were living tissues, those were made up of cells. They still are made up of cells. They happen to be dead now. Well, there's DNA in them, there's RNA in them. So we need to be able to break down that plant animal DNA and RNA into sugars, phosphates, and bases. And how do we do that? Enzymes. We can't live without them. So the basis of most of your digestion, most of your chemical digestion, relies on enzymes. Enzymes speed up chemical reactions by lowering the activation energy. What they do is they put a strain on a bond and make that chemical bond easier to break, which is exactly what we're doing when we're digesting food. You're taking big polymers, big macromolecules, a big strand of DNA, or a big lipid, or a big protein, or a big polysaccharide, complex carb, and you're breaking it down mechanically and you're breaking it down chemically. Enzymes break them down chemically by putting strain on the bonds, breaking the bonds more easily. So we're literally chopping up those big macromolecules into monomers, into building blocks. Okay? And remember, enzymes are reusable. You can use them over and over again as long as they're not damaged by heat or damaged by radiation or damaged by big changes in pH. All right?